As we marvel at the capabilities of CRISPR, we find ourselves on the precipice of a new age, an era where the very essence of life, its building blocks, are at our disposal. Yet, such power comes with its share of responsibilities, a point that we must remember to remain consciously aware of as we stand in awe of the possibilities that this genetic tool presents. Every groundbreaking innovation, like CRISPR, is invariably accompanied by a myriad of considerations that necessitate thorough exploration. There are potential risks to discern, ethical dilemmas to solve, and societal implications to evaluate. Let's delve deeper into what these entail, starting with the scientific risks, primarily the possibility of off-target effects. Off-target effects, a phrase that in the context of genetics bears substantial implications. The term might seem nebulous to those unacquainted with the complex world of genetics, but its meaning is straightforward yet profoundly significant. Off-target refers to the unintended alteration of genes, an accidental genetic modification that happens as a result of the Cas9 enzyme's action, an enzyme that forms a fundamental component of the CRISPR system. Despite CRISPR's lauded precision, there is a residual probability, no matter how microscopic, that Cas9 could deviate from its intended course. A slight error, an almost insignificant miscalculation, could result in the enzyme inadvertently modifying an unintended genetic sequence. The ramifications of such a misstep can span that Cas9 could deviate from its intended course. A slight error, an almost insignificant miscalculation, could result in the enzyme inadvertently modifying an unintended genetic sequence. The ramifications of such a misstep can span the spectrum from innocuous to downright catastrophic. To understand why a seemingly minor error can have such profound consequences, we must appreciate the interplay between genes. A gene isn't an isolated entity. It interacts with other genes and the environment in a complex network of cause and effect. A change in one gene can inadvertently trigger a cascade of changes in others, setting off what could be best described as an unexpected chain reaction, a domino effect of genetic modifications. To visualize this, imagine an elaborate arrangement of dominoes, perfectly balanced and meticulously aligned. A single nudge to one domino can instigate an entire sequence to topple, in a spectacle both mesmerizing and unnerving. Now consider a scenario where each domino represents a gene within the intricate structure of the human genome. The off-target effect of CRISPR could be likened to mistakenly nudging the wrong domino, potentially chain of events within the genome. The resulting health implications or diseases could be unforeseen, unanticipated, and challenging to address due to their complex and interconnected nature. But the concerns surrounding CRISPR don't end with scientific risks. We also find ourselves face to face with substantial ethical conundrums that CRISPR's application brings to the fore. Central to these is the controversy embroiling the concept of designer babies. This notion, once relegated to speculative science fiction, has been thrust into the realm of feasibility, thanks to the leaps and bounds made in genetic engineering. CRISPR opens the door to a future where parents can handpick specific traits for their offspring, essentially tailoring bespoke genetic profiles. While this may initially strike us as an exciting and revolutionary prospect, further contemplation reveals a darker underbelly. The ability to genetically customize offspring eerily echoes the principles of eugenics, a field fraught with ethical and societal concerns. The fear that looms large is that such genetic enhancements could become the privilege of the affluent, thereby deepening the chasm between the haves and the have-nots. A new form of disparity could emerge, wherein only the rich can afford genetic superiority, creating a stark genetic divide that mirrors and intensifies socioeconomic inequalities. We must also consider the contentious issue of consent. CRISPR makes it possible to modify an embryo's genes, effectively making life-altering decisions for an individual unable to give their consent. This raises intricate questions regarding personal autonomy and physical integrity. Philosophers, ethicists, scientists, and even laymen grapple with these ethical dilemmas, searching for a consensus on the boundaries that demarcate the ethical from the unethical in this brave new world of genetic manipulation. In this powerful tool with the utmost caution, we bear the responsibility not only from a scientific perspective, but also an ethical and societal one. We must navigate this path with a clear understanding of the immense responsibility in our hands and the ethical consequences that we face from the vast changes this could bring. After exploring the potential horrors and ethical dilemmas of CRISPR, we now delve into some specific issues that have sparked intense debate and concern. One such issue is germline editing. Germline editing involves making modifications to sperm, eggs, or embryos, changes that are heritable and can be passed down to future generations. This raises significant ethical questions, primarily around consent and long-term unforeseen effects. While germline editing could potentially eradicate certain genetic diseases, its misuse could lead to a dystopian future where genetic traits are selected and modified at will, leading to a designer baby scenario. Next, we move on to the topic of gene drives. Gene drives created using CRISPR can promote the rapid spread of genetic modifications through a population. This could be beneficial for controlling disease vectors like mosquitoes, but what about the unintended ecological consequences? Could we accidentally trigger an irreversible change to an entire ecosystem? Then, there is the concern over biodiversity. The introduction of genetically modified organisms, GMOs, created using CRISPR into the wild could have unpredictable effects on natural ecosystems. The balance of nature is delicate, and meddling with it could lead to unforeseen ramifications. And finally, we must consider the regulatory frameworks, or more accurately, the lack thereof. Different countries have varying regulations regarding CRISPR technology, leading to a lack of international consensus. This poses challenges for global collaboration and governance. The need for strict oversight and ethical review of CRISPR research, particularly when it involves human subjects, has been emphasized by governments worldwide. CRISPR also falls under the purview of various international agreements, such as the Cartagena Protocol on Biosafety, which aims to ensure the safe handling, transfer, and use of living modified organisms resulting from biotechnology. Furthermore, international declarations, like the Universal Declaration on the Human Genome and Human Rights by UNESCO, provide a framework for ethical considerations in genetic research, which includes CRISPR technology. In this scene, we will explore these concerns in greater detail, examining the potential consequences of each, and seeking to understand how we might navigate this ethical minefield. As we navigate the complex labyrinth of CRISPR's implications, we must also acknowledge its potential as a life-saving tool in times of crisis. One such instance is the COVID-19 pandemic, a global health emergency that has challenged the scientific community like never before. The urgency and scale of the pandemic have prompted researchers worldwide to explore every possible avenue to combat the virus, and CRISPR has emerged as a promising contender. In the early stages of the pandemic, CRISPR technology was explored for several purposes. The first of these was in the realm of diagnostic tools. CRISPR-based tests, such as the Sherlock and Detector systems, were developed to detect SARS-CoV-2, the virus causing COVID-19. These tests aim to offer a quicker, simpler, and more accessible testing solution compared to traditional PCR methods. The rapid and accurate detection of the virus is a crucial step in controlling its spread, and CRISPR's potential as a diagnostic tool cannot be overstated. CRISPR was also instrumental in research to understand the genetics of the virus and how it interacts with human cells. By using CRISPR to manipulate the genes of the virus, scientists could observe how changes in the virus's genetic material affected its behavior. This insight into the virus's workings is invaluable in developing effective treatments and vaccines. The third application of CRISPR during the pandemic was in exploring potential therapeutic approaches. While CRISPR-based treatments for COVID-19 were not readily available at the onset of the pandemic, the technology held promise for future therapeutic strategies. These strategies could involve directly targeting viral genes or modifying host genes to confer resistance to the virus. However, even as we harness the power of CRISPR to combat global health emergencies, we must tread carefully. Governments and regulatory bodies worldwide have expressed apprehension about the rapid advancements in CRISPR technology. Their concerns primarily revolve around the ethical, safety, and environmental implications of CRISPR's widespread use. This cautious approach is crucial to ensuring that we wield this powerful tool responsibly, without losing sight of the potential risks and ethical dilemmas it presents. In the next scene, we will delve deeper into these concerns and explore how we can navigate the ethical and societal implications of this groundbreaking technology.